So Diego Ferreiro is a professor of biological chemistry at the University of Buenos Aires in Argentina. He was trained as a biologist and got a PhD in biochemistry at the University of Buenos Aires. And then he went to do his postdoc at UCSD. And I hear he overlapped with Akif a little bit while he was there. Diego, since 2010, runs a protein physiology laboratory in University of Buenos Aires, and he studies everything about proteins, sequence, evolution, domains, all the relationships between these things. In fact, when I was introduced to Diego in 2016, he was introduced to me as a protein guru. And some of the things that he does, I talked about today. His earlier work allowed for recognition of these repeat substructures, these tiles in our central spheres that would have been present on the origins of life. And today, Diego is going to talk about these repeat substructures more, but more about their dynamics and folding. So Diego?
Well, thanks for the invitation. I'm sorry I couldn't do it in person, but at least we have this opportunity to be remote from different parts of the world. Also, excuse me because I'm a little bit sick, probably with coronavirus. That's why my voice is in this tone and my ideas are a little bit fuzzy. So today I'm going to present mostly the work of Ezequiel Galpern. Ezequiel is really a brilliant PhD student in our lab, and he has been developing this work in collaboration with Alexandra Thierry and Jacopo at the École Normale Supérieure, which are experts in statistical mechanics. And we work together at the Departamento de Química Biológica at the University of Buenos Aires. So when we think about evolution in proteins, we think about how's the information flow in proteins. So we have these sequences, which are a way of digitally coding information that are expressed and in some short time scales in a given environment, they give us these structures. These structures in turn interconvert, making up some dynamics that we can define that is usually in between the millisecond to the gigasecond time scales that give these activities. And in turn, the activities come together from different proteins and from proteins interacting with the whole of the rest of the stuff they find around in a time scale of milliseconds, again, to years, to give up biological functions. And here is where the classical central dogma of molecular biology ends, going from sequences to structures to functions. But of course, we are missing a central thing that has been talked about, and this is quite a hot topic that has been shown to be very useful in analyzing protein sequences and structures. That is evolution. Basically, there is a feedback of information from the environment to the genomes in that usually it's separated in time scale. It goes into the thousands to the millions of years. But as Chris Adami pointed out almost a decade ago, is that we have to think about evolution flowing, information flowing from the environment to the genomes. And here in the ENIGMA paradigm, we have to take care that the protein's activities themselves modify the environment. So again, we are, as Professor Bental told us, in a chicken and egg problem because we have this circular flow of information. When we see the structures of proteins, the modern proteins, these are highly evolved objects. Here are just these aquarels or water paints that David Goodsell does around the high resolution structures of proteins. And we see they are formed with subunits, and subunits can be decomposed into domains. and probably domains can be decomposed in terms into something smaller that we are still not agreeing how to call them. Someone call them Lego blocks, someone call them tiles. So, Professor Bental introduced themes, motifs. So there is something that goes below the domain, but higher than the amino acid, that is fundamental to protein structure definition. But when we see the proteins today, we have to take into account that they're highly evolved, and most of them, the interaction between the building blocks is highly tightly regulated and optimized. Ancient proteins must have been simpler systems. And I will claim here, and I've been claiming before, and it's not only myself that think about this, is that likely are repeat proteins, that repetitions, the fact that you have repetitions may lead to building up symmetric arrangements. And this makes the assemblage to be easier from building blocks. So larger objects, functionally, probably functional, larger objects can be built from simple structures. In turn, there are theoretical arguments emerging from energy landscape theory that the symmetric proteins are easier to find by random search in the sequence space. So it's easier to find sequences that fold if you have repetitions. And in turn, this is there is another argument that having repetitions can be a way of doing an efficient use of a limited coding capacity. So if you have a small genome, you can code for one repetition and then just make copies of the same repetition to build something larger that comes together. So the big problem here is how do these proteins formed with repetition fold and function? We have claimed that the functional patterns are the ones that modulate the genomic sequences. So can we actually look at patterns in sequences and use them to infer something about folding? Well, I would show you that we can do this at least for some of the systems we study. And we are using the most simple example we have of natural repeat proteins, and are the class of ankyrin repeat proteins. This class of proteins are ubiquitous. They are found in all the living organisms, and they are made of tandem repetitions of a motif of 33 amino acids, around 33 amino acids. Of course, not all repetitions are the same. You have variations in length, and you have variations in the composition, and you have variations in the number of repetitions, but people have been working with these proteins, and they have shown, for example, that if you synthesize one repetition, so if you synthesize a peptide of 33 amino acids, it doesn't fold. If you synthesize a peptide with 66 amino acids, having 2 copies of the repetitions, it doesn't fold. If you synthesize a protein of 3 copies of repetitions, it does fold under some conditions. And if you synthesize a protein of 4 copies of this particular sequence of amino acids, it does fold, and you can solve the crystal structure. And in the crystal structure, you can see that each of the repetitions adopt a repeating pattern. And in fact, the repeating part interacts by near neighbors with the rest of the protein. So we see this, we analyze the folding of this object, and we find it actually folds either all the repetitions folded or all the repetitions unfolded. It really behaves as one domain, but not as any single repetition by its own faults and functions at one domain. So we have a problem here if in the identification of the elements that repeats themselves and how they come together to form domains. Unfortunately for the ENIGMA theme 1, this is not an example of a metal binding protein, although there are some examples of these members of this family that do bind metals, and there are several examples of proteins that do bind other things. We treat them together and we try to average out, we expect to average out the functional information to keep only the folding energetic information, which is the thing we believe is common to all these objects. So we want to predict how the protein really folds, basically the repeat protein folding mechanism. We want to predict the stability of the protein, the cooperativity of the folding units, if they are intermediates, and so on. We want to use only sequence information. By this, I will show you that it's enough to look at the local frequencies, the pair frequencies, and the length of the protein and the identity between consecutive repeats. And we want to do this on a large scale. Large scale, we want to analyze the folding of thousands of proteins. So we propose a general pipeline for which we need a large collection of sequences aligned in a multiple sequence alignment. From this, we will show you how to derive an evolutionary field, basically direct coupling analysis, custom made for repeat proteins. And with this, we will try to map the information at the single sequence level to a coarse-grained description of a folding model, for which we will take some part of the protein, some motif of the protein, some theme, maybe some repeat or sub-repeat of the protein, and assign a folding probability, and we will define interactions between them. Of course, we will be building and looking at spherical cows. These are not proteins, certainly, but we will try to see how far we can make the cows go, because this can be a very good model of a cow if we are comparing it to zebras or to bacteria or to other living objects. So this might not be a terribly bad model to analyze protein folding at the large scales. So the simplest model to account for the sequences are some observables. We will look at the multiple sequence alignment. We will look at the frequency of occurrence of each amino acid in each position. We will look at the joint frequencies between every pair of sites, basically the frequency of occurrence of amino acids in position A and position B together. And we will look at the identity of consecutive repeats as another sequence observable. And we will take into account the length distribution of the proteins. I told you there were proteins with 2, 3, 4 repeats, but there are proteins with 20, 40, and even 100 repeats. And we will take for this a mechanistic model for repeat duplication and deletion. People have been analyzing that repeat proteins not only evolve by point mutations, but you can identify in the existing proteins repeat duplications and deletions. So for this, we will build up a model that has we will say that the probability of finding a particular sequence is a Boltzmann distribution of some energy. This energy can be obtained by a maximum entropy principle and can be derived in local fields which is basically related to the frequency of one amino acid and one position, and the couplings between every pair of amino acids in every pair of positions. And we fit this with a Boltzmann learning. Boltzmann learning is a very expensive way of coming up with parameters for the J's and the H's. But we found that for these repeat proteins, we have enough information to actually do this without overfitting. And they actually find that the fitting is robust and it converges without overfitting. For this, we need about 150,000 sequences. One thing we can immediately look at is the JIJs, basically the interaction potential between every pair of amino acids. Here in red, you see here it's two repeats, two consecutive repeats. Remember, each repeat has 33 amino acids. So it's 1, 2 repeats versus 1, 2 repeats. The red points are the highest JIJs, basically highest interactions, and the gray is the contact maps of one known experimental structure. This is not strictly novel. We are just recovering interactions, physical interactions from the sequences. The PID distribution, basically the distribution of identity to first neighbors, is reproduced. And also the 3-point correlation frequencies are reproduced. This is the model 3-point correlation and the data 3-point correlation. And it's distributed. So this is not that we are particularly overfitting the data. Notably, the changes in the evolutionary, we call this evolutionary energy, the changes in evolutionary energy that we can compute for a protein and a mutant is related to the change in the folding free energy of the mutants and the wild type protein. So you can take, for example, the I kappa B protein, which is an ankyrin repeat protein, and experimentally do one mutation or a mutation of more than one amino acids, do the folding experiment on I kappa B, measure the free energy change from going to folded to unfolded, and then measure the difference in the free energy change for folding for the mutant. And this will give you one data point with one error bar. And you can do this for a bunch of mutants on a bunch of proteins, and we can compute this directly with this evolutionary energy, because any sequence will have an energy assigned to it, which just comes from the actual amino acid distribution. This is a very good correlation, even though for some, if you're not familiar with this kind of data, it might look very noisy and not a good correlation. This is even better than the correlation you will get for other methods that try to estimate free energy change upon mutation applied to these proteins. And we will use this, that we believe has something to do with folding, to try to map this evolutionary energy to a folding model. So how do we do this? This is the evolutionary information of one particular protein. So UniProt ID P44771 is one protein for which we have a three-dimensional structure. And this is the sequence of this protein. And here are the interactions between every pair of amino acids, basically the JIJs that come after fitting, and the H's are on the diagonal. And we will say that a bunch of consecutive amino acids, in this case, this part of amino acids, which correspond to these amino acids, will make one folding element. This folding element, sigma 1, will have an energy that is just the sum of the energies of all the points in this little square. Will make this big square. Then we have to do the same for sigma 2, sigma 3, sigma 4, and so forth. So we have these sigmas, these energies assigned to each sigmas. And then for every pair of interacting elements, we will assign a surface energy, which will be these cases, these cases, these cases, these cases, and so forth. So we will fill in a matrix for which we can define a model in which we will have the energy of the Hamiltonian of this model being such that each of the elements will have an energy if it's folded, or it will contribute an entropy if it's unfolded. And if two elements are interacting, when they are folded, they will contribute a surface energy. This is basically what physicists call an Ising-like model. So we are mapping a Potts-like model, which is the evolutionary model, to an Ising-like model, which is the folding model. This is the coloring of the structure corresponds to the assignation of the elements. We did this assignation of the elements based on the secondary structure of the these particular systems. So we look at some of the proteins for which there is experimental data. This is the same protein we treated before. P16 is a 4 ankyrin repeat protein. These 4 ankyrin repeats correspond to 8 elements, 8 folding elements. And here is the experimental data of the thermal unfolding of this protein. And this is the simulation data we got for the thermal unfolding of this protein. This is a rather good fit. And how is this protein folding? Well, we can actually draw, for example, a free energy profile of the folding of the elements. So if you are at a very high temperature, the free energy is lowest when you have 0 elements folded. As you lower the temperature, this free energy changes go to this place, and you find a minimum of 4 elements folded. Notice that it goes from 0 to 4 elements folded at the same time, and then it folds 5, 6, 7, and if the temperature is low enough, you find the 8 elements folded. We can look at the correlation of the folded elements, and we see that element number 4, 5, 6, and 7 together make the transition. It's basically this jump from 0 to 4, and then element 3, 2, 1, and 8 fold independently of elements 4 to 7. This is drawn here in the colored accordingly in this three-dimensional representation. And we did this kind of analysis for all the proteins for which there is experimental data, trying to fit the only free parameter. Remember, the internal energies and the surface energies are given by the sequence. We only have the free parameter of the entropy contributed by each element. And here are the fits of 6 different proteins and mutants for which the dots are the experimental data and the lines are the simulated data. we see that there is a reasonable good agreement, not with all of them. But if we look at how well this is fitted based on the scanning of the entropy per residue, we find that for all of the systems in between 4 and 6 calories per Kelvin mole is a good value of the entropy per residue, which is in line with previously estimated configurational entropy losses per folding. So we were very happy to see this. And then we have a tool to actually go and look for the folding of thousands of proteins. We analyzed the folding of 4,000 ankyrin repeat proteins, for which we derive the energies from the sequences and the entropies from just setting a common value of 5 calories per Kelvin mole. And we find several different cases of behaviors. Say, for example, this protein. So this is the UniProt code, and these are the residues from numbers 178 to 309. This is the simulation of the folding, fraction folded versus temperature. Everything is folded, and then suddenly everything is unfolded. Here we see the correlation of the folding of the whole protein. And here we see the free energy plot. So as you can see, this protein is either everything is unfolded or everything is folded. Basically, the whole protein undergoes a two-state transition. This is called two-state, everything unfolded or everything folded. There are some examples for which we see this kind of behavior, in which the protein undergoes a folding element by element, in which the elements are not correlated at all. So this is folding one element by one element, which we call a downhill folding, because there is no free energy barrier for folding this protein. There are some examples of proteins that undergo several jumps, in which we see some state and some minimum of free energy, then a high barrier and some other bunch of elements folded, and then another jump to some more elements folding. And we can correlate this in the sequence and find that there is a nucleus that folds, and then it just jumps and folds another bunch of elements, which we can call a domain, and another bunch of elements still at lower temperatures. This is reminiscent to an intermittent folding, in which if you have this protein, you will have 3 transitions. Or we find examples like this one, in which we see some downhill folding, then a free energy barrier, and then some other downhill folding. And we can identify these domains emerging from this particular sequence. So basically, there are very different mechanisms, even if you have the same topology. Remember, these are just the same ankyrin repeat proteins. Even in the same topology, you can find out there are very different folding mechanisms. We can do and look at for the if the domains that we identify, how this distribute in the 4,000 proteins we analyze. Basically, there is in between 1 and 10 domains per array. There is a slight variation in the number of domains as you increase the size of the protein. This is the size of the protein, and this is the average domain size. So for all the domains, there is not a big variation. But if we look at the first domain to fold, basically the domain that falls to higher temperature, we see there is a correlation of about 0.5. So basically the nucleation of the folding of the array encompasses about half of the protein. So you have the full protein unfolded, an array of consecutive, about half of the elements fall together in a lot of the proteins. We can look at the cooperativity and the stability. There is a linear relationship between the normalized energy, which can be computed directly from the sequences, and the folding temperature of that sequence. So basically the stability can be predicted directly from the energy. We don't have to do the simulation. And the cooperativity, well, the cooperativity is not so easy to find the descriptors, but Ezequiel was clever enough to look at two kinds of description of the energies. Basically, he looked at the heterogeneity of the energy along the array, basically how different the energy is distributed along the array of repeats, and how strong are the interactions between the repeats. And he finds this distribution for the 4,000 proteins. So notice that the proteins are either very cooperative, all or none transition, or very uncooperative, intermittent or downhill folding. See, there are no proteins here for which there is a high heterogeneity and high interactions. It's not the case. You can have high heterogeneity or low heterogeneity with low interactions, or you only have high interactions when you have low heterogeneity, and the cooperativity can be more or less predicted from the variations in these energy terms. Moreover, and to end this, I don't know if I'm very much over time, the actual evolutionary field is generative. We can actually do a Monte Carlo run on the field, and you can come up with sequences. And if you try to distinguish the natural and the simulated sequences, say, for example, by building a pairwise sequence comparison, you cannot distinguish one. We haven't found any particular way of distinguishing a simulated sequence from a natural sequence. So this opens the possibility for us to actually simulate sequences and simulate the folding of these sequences. And again, we can come up with this kind of a plot in which we look at the cooperativity versus the energy variations. And we can actually design, for example, these 13 proteins for which we predict the folding temperature of each of the elements. So here is the folding temperature and the cooperativity for a bunch of proteins. And we are actually in the process of synthesizing this protein and actually look if this is true, if we can come up for proteins that have the same fold, the same overall structure, but very different cooperativity and stability. And we could actually look at evolutionary trajectories. We can look for, again, a Monte Carlo simulation and look for the energy of sequences, and we see this kind of jumps. At some particular temperature, we see that the sequences are either random looking or they are look like anchoring repeat proteins, or they are random looking at the anchoring. So this is reminiscent of what Hyu-Sui Chan proposed in 2014, in which you have the sequence space distributed as a neutral network, and you have the different topologies acting as attractors. So if we can build up evolutionary fields for different type of repeats, we could actually begin exploring how the sequences get to change over evolutionary times. So can we actually explore the evolutionary dynamics? I think it's an open question now. So as a summary, we developed a coarse-grained folding model of repeat proteins that recapitulates the folding dynamics. The folding energetics can be derived only from sequence information. This model can predict experimental observables, such as thermal folding curves, folding domains, free energy profiles, etc. The stability and the cooperativity of the repeat arrays can be explained by these evolutionary energy terms. And this field is generative. So we can actually specify different sequences that are predicted to have different folding experiments. We are doing these experiments. And what we actually don't know if we can use these models to actually explore the evolutionary dynamics. I really don't know what would be the experiment we can do here and how to test this. All of this data is deposited actually in an archive paper by Ezekiel and ourselves. And there are the GitHub also codes for all the runs and how to simulate the folding and so. So I'll be happy to take questions. Thank you.
Questions? Go ahead. You get a speaker right here.
Hi, so I'm not part of the protein world, but it seems like you have so much thermodynamic data here. You have not only energies at the completely folded and completely unfolded states, but you have energies at the maxima, energy maxima that go through intermediate states that would basically constitute an activation energy of folding. And I wondered, has you or anyone else done experimental kinetic experiments, and do those match the activation energies that you would predict from these models?
Wonderful, fantastic question. So one answer is yes, there are experiments, and actually myself, we have done some experiments about the actual activation energies for ankyrin repeat proteins, but we didn't actually look at how these activation energies match the experimental ones, except in one only system, in one system only, for which we looked at this. This is the actual system for which we look at the matching between the perturbation of interactions in the protein at some particular regions and the effect of these regions to the activation barrier. Basically to see if this block that we are seeing here, it actually matches the experimental block that has been observed. This is called usually phi value analysis. It's basically looking at the effect of mutations in the activation step relative to the thermodynamic step.
Well, that seems like a fun area to explore, to find out, are there styles of folding that are particularly easy once you see the energy barriers?
Absolutely. Here we have a prediction, say for example here, the kinetic of folding of this protein should be absolutely fast. This has no barrier compared to this one that has a barrier of about, in this case, 5 kBt, but can be higher.
Thank you.
Welcome.
Did you have a question?
Hi, Diego. Great stuff as always.
Thanks, Vic.
Two small questions for you. One is, we think about the size of protein domains as being around, I don't know, 200, 300 amino acids. And a lot of that is that if it gets smaller than that, then you don't have enough of a core. And if it gets larger than that, then it gets unwieldy to fold as a single cooperative unit. I wonder if you can see that limit theoretically, because you can model a two-state folder. How many repeats can you build in before that two-state folder starts to fall apart? And then the second question is, in natural repeat proteins, you see this phenomenon where adjacent elements, adjacent folding elements, have very different sequences from each other to prevent crosstalk and misfolding. Is that something that's encoded in your model, or do you only have adjacent interactions between units? Thanks.
No, thank you. First question. First question is, we didn't do experiment. Nice calculation to be done, basically to look at the smallest folding unit. We don't know, but in the case of this topology in particularly, we see that the experiment has been done, and it's 3 repetitions, so it's 99 amino acids. I will say that you can have folding domains of about 50 amino acids, which already can have a folded core, but we haven't done the calculation of what the smallest core can be. And actually it would be tricky because we will have to find the most energetic element. So basically we'll have to define which is the better folding element and then put them together until we get a reasonably well stable system. I don't know, we haven't done the calculation. And the next question, it was related to, can you repeat it?
So the
Sorry,
second
you have to
question
come
is,
up.
you have in repeat proteins, adjacent elements often have very
Yeah,
different sequences.
okay, the
to prevent misfolding.
crosstalk. I remember. No, so this is, in our model, there is no crosstalk, as you can see here. So the crosstalk will be the interactions between elements that are distant in sequence, right here, around this area of the matrix. This is not encoded in our model. As you see, these elements have 0 interaction. And besides, we have no frustration. You see, there's only one place we can go. There is only one structure this can form. It doesn't have the alternative structures. But I can tell you that we have done some folding experiments, folding simulations with a more detailed energy interactions and molecular dynamics, in which we allow for interactions between distant elements. And we actually do find some cross domain swapped, basically let's call them domain swapped traps, but they're not very common, particularly in these topologies. And this can be explained because they are very cooperative. Once you have a nucleation of a couple of repeats, it's very easy to actually fold upon the already formed nuclei. It's much more difficult to pay the entropy cost of folding distant elements. So if you include this topological frustration between distant elements, you do find the traps, but they are not very favorable. It is much more favorable to go to the native state than to the trapped states in this particular topology.
Thanks.
Welcome.
Okay, cool. I have a question, but I think we're over time for a break. So if you guys want to go ahead.
15-minute break.
Oh, break, yes.
Here at about 25 after.
Diego, I will ask you my question anyway. Uh,
Thanks to everybody. I really want to thank you for doing such an impressive job of putting this all together. So well.